Bismillahirrahmanirrahim. Assalamu alaikum dear all students of Fundamentals of Medical Laboratory Technology. Today we will discuss about the history and taxonomy of microbiology, which is one of the major domain, the major part of medical laboratory technology. So this is the lecture in which we will discuss about the introduction to microbiology, that what is microbiology, what are the major divisions of microbiology, and then we will discuss about the different types of prokaryotes and eukaryotes, the microorganisms which are divided on the basis of the cell functions and structures, and then we will discuss about the scope of microbiology, that where microbiology is applied, what are the implications, what are the applications, what are the implementations, and what is the scope of microbiology. Then we will discuss about the history of microbiology, that how microbiology subject and the related history developed, and then we will discuss about the taxonomy of the microbiology. So this is the outlines of the today's topic. The introduction, the first outline, the first part of the today's lecture. Microbiology is basically the study of the microorganisms, and microorganisms is splits into micro and organisms. Micro are those organisms which are too small to be seen with the naked eye, or these are the organisms which cannot be seen with the naked or unaided eye. So therefore these organisms are called microorganisms, and this micro is defined as any organisms which have diameter usually less than 1 millimeter, or the organisms which cannot be seen with the eye without any instrument, without any help. So this was the introduction about the microbiology. The microbial taxonomy, as we know that the microorganisms is found everywhere in the universe, and microorganisms are very diverse, and the scientists, the taxonomists, they always found challenging to classify the microbes, the microorganisms in the various environments, in the various niches, in the various areas of the universe and the ecosystem. So on the basis of microorganisms which causes infectious disease in human, so the microorganisms are divided into 5 different classes, and these are the major groups of microorganisms, the major classification of microbiology. So bacteria are the unicellular, the prokaryotes, then the second one is the fungi, the third one is protozoans, the helminths and the viruses. So these are the 5 major groups of microorganisms in which microbiology and microorganisms is divided, and these microorganisms have relation with the human in case of any infectious disease, or in fact in case of the living relation, in combination having the part of the normal flora of the body, the human, or they cause the infections with the humans. So therefore these are the 5 major classification of microbial taxonomy. The 4 different classifications, the 4 different kingdoms in which the animal kingdom, Animalia, the pathogenic microorganism is basically the worms, and the cell they contain are the eukaryotic cells. The protist the example of the protist is the protozoans, which are basically eukaryotes, and the fungi, kingdom fungi, in which the fungi, the yeast or the molds, they causes disease in humans, and their cell type is also eukaryotic. The prokaryotes is divided into bacteria and viruses, in which the bacteria is prokaryote and the virus is a non-cellular microbe. So, this is the classification of the pathogenic microorganisms and their type of cells. Now, what is prokaryote and what is eukaryote? How we can say that this type of cell is prokaryote and this is a eukaryotic type of cell? So, the prokaryotic organisms have a very simpler cellular morphology, and these type of cells do not have the organelles which are membrane bounded. So, therefore, these cells and these type of organisms are called prokaryotes, and the eukaryotes, they have all the cellular components which are bounded by the membrane, which include the nucleus, and they are usually complex than the prokaryotic type of cells and have larger membrane, and the size is larger than the prokaryote. This is the difference between the prokaryotes and the eukaryote. So, the first characteristics is the DNA. The DNA within a nuclear membrane is present in the eukaryote, but the DNA or the genetic material is found in the prokaryotes, but they are not enclosed within a nuclear membrane. The mitotic cell division, it is found in the eukaryotes. Eukaryotic cells are the human cells, but it is not present in the bacterial cells. So, the bacteria divide by binary fission, and the DNA associated with histones is absent in the prokaryotic cell, but it is present in the eukaryotic cells. The chromosome number, the bacterial chromosome number is 1, but in the human eukaryotic cell there are more than 1 chromosomes. The membrane bounded organelles, for example mitochondria, lysosomes, these organelles are absent in the bacterial cell, which is a prokaryotic, and present in the human cell, a eukaryotic type of cell. The size of ribosomes is also smaller in the prokaryotic bacterial cell, which is overall 70s, the larger subunit 50 and the smaller subunit 30s, whereas in the eukaryotic cells the overall size of the ribosomes is 80s. The cell wall containing peptidoglycan, it is present in the prokaryotic bacterial cell, but cell wall containing peptidoglycan is absent in the eukaryotic human cell. Now, the different divisions of microbiology, the bacteriology, the study of the bacteria and the diseases which is caused by bacteria is called the bacteriology, and the virology, virology is the study of viruses and the diseases, the conditions associated with the viruses. So, this type of study is called virology. The third division of microbiology is the mycology, and mycology is the study of the fungi and their associated disorders, their associated infections and the conditions. So, the fourth one is the parasitology, the study of parasites, the infection, the diseases, the association of any condition with the parasites and their study, their finding. So, this is called the parasitology. The important characteristics of the viruses, bacteria, fungi, protozoans and helminths. So, the first we will discuss about the cells. So, viruses are those organisms, are those microbes which are not cellular, but they are usually the infectious agents and the infectious particles. The bacteria contain also cells, the fungi and the protozoan and helminths also have cells. The diameter of the microbes, the viruses diameter ranges from 0.02 micrometer to 0.2 micrometer. The bacteria are slightly larger than the viruses and their diameter ranges from 1 to 5 micrometer. The fungi is slightly larger in size than the bacteria and their size is 3 to 10 micrometer. Example, especially in the yeast are larger, and the trophozoites, their approximate diameter is 15 to 25 micrometer. The nucleic acid, they contain, the viruses either contain the DNA or the RNA. Only one type of nuclear material may be present in the viruses, but in the bacteria, the fungi and the helminths, both DNA and RNA both are present and both are must for their survival. The type of nucleus, so as we know that the virus is a non-cellular, so there will be no nucleus in the virus, but the bacteria is cellular, but their nucleus is simpler one and it is not bounded by a membrane. So, therefore, they have a prokaryotic type of nucleus, whereas fungi and helminths and protozoans, they have the eukaryotic type of nucleus. Ribosomes is absent in the viruses and ribosomes are present in the bacteria, but their size is smaller as compared to the fungi and helminths which have 80S, whereas bacteria have 70S. The mitochondria is absent in the viruses and bacteria, but they are present in the eukaryotes, which is the fungi, the protozoans and the helminths. The nature of the outer surface. So, basically the virus have a outer capsid protein and a lipoprotein envelope, whereas the bacteria have rigid cell wall containing peptidoglycan, the fungi have rigid cell wall containing chitin, and the protozoan and helminths have flexible membrane. So, all the microbes, they have different types of cell membrane. The motility is absent in the viruses, where some bacteria are motile and certain bacteria are non-motile. The fungi are overall non-motile, whereas the protozoan and helminths, most of them are motile. The method of the replication. So, for replication, the viruses depends on their host machinery and they are not replicated by the binary fission. The bacteria, they replicate by binary fission and the fungi replicate by budding and in certain cases mitosis, whereas the protozoan and helminths, they replicate by the process of mitosis. The scope of microbiology. Microbiology and the microorganisms are found everywhere, from the geothermal vents into the deep oceans and from the to the coldest Arctic ice to every person's skin. So, microorganisms can be found anywhere in the universe, and it is estimated that microbes contain 50% of the biological carbon and 90% of the biological nitrogen on earth. So, the importance and the scope of microbiology can be observed by reading these numbers, that the most of the nitrogen that is present and that is contained within the microorganisms, and the microorganisms are the major contributing factors to the functioning of the biosphere, to the functioning of the universe, and above all of them, the microorganisms are the source of nutrients, and they are the initial source of all of the ecological food chains and food webs. So these have a role in every part of life and in everywhere in the universe and in every systems which is relating to the life. But the microorganisms carry out photosynthesis, so they are rivaling the plants in their role of capturing carbon dioxide, and they release oxygen into the atmosphere. And certain microbes inhabit human, so they make a major part of the normal flora of the human and have important role and helping the body immune system, as well as they help in the digestion of food, and within the gut of human they produce different type of hormones, for example vitamin D, vitamin B and vitamin K, which have important role in the clotting mechanism and the preparation of multiple clotting factors. The microbes are important in the production of bread in the different food products, for example the cheese, the beer, the antibiotics. The microbes have role in the production of antibiotics. Certain microbes can be used as antibiotics, and they have also role in the vaccine, in the production of vitamins and production of enzymes and many other factors, many other products which have been prepared by utilizing these microbes. And all these mechanisms, all these products are produced under the biotechnology and their related projects, because the biotechnology rests upon its the biotechnology rests upon a microbiological foundation. So in 1347, the plague, or also called the Black Death, which is an arthropod borne disease by Yersinia pestis, it struck the Europe with a very brutal force and killing about 1/3 of the population, which counting about round about 25 million of the people, and that was within the 4 years after the attack. So during these 4 years, this Black Death or the plague, they killed more than 25 million people. So in the next 80 years, the disease struck again and again. The more outbreaks of the disease appeared, and they killed round about 75% of the European population. So the plague have a very bad effect, that some of the writers of the history, they believe that this plague, this disease changed the European culture and prepared the way for their Renaissance period. The history of the microbiology: before the microorganisms were seen, some investigators, they believe, they suspected that there are certain agents which exist and they are responsible for multiple types of diseases. A Roman philosopher, Lucretius, from 98 to 55 BC, and another physician, Girolamo Fracastoro, from 1478 to 1553, suggested that diseases were caused by a certain specific type of creatures which are living but they are invisible. So these invisible creatures have role in the developing of a disease, and the earliest microscopic observations, they were prepared, they were made in 1625 to 1630, on bees and weevils by an Italian microscopist Francesco Stellucci using a microscope which may be probably provided by the Galileo. So, in the 1665 the first drawing of microorganisms was reported in Robert Hooke Microphagia. This drawings shown different type of bacteria and their structures. The first person who published the accurate observation of microorganism was the microscopist Antony Leeuwenhoek, and the microscope he prepared could magnify the microorganism from 500 to 300 times, and he may have illuminated his different type of specimen which are converted into liquid by placing them between the two pieces of glasses and shining light on them at the 45 angle, so to the specimen plane. The Leeuwenhoek observations were the development of microbiology and essentially linguist for the next 200 years. So, this is Mr. Leeuwenhoek. This is his microscope. So, the lens of the microscopes, the specimen holder, this is the focus screw which are used for the focusing, and this is the handle. So, these are the first figures. This is the which he prepared after reporting the different type of bacteria and their structure. So, this is a simple bacteria, this is a chain shape of bacteria. So, these are the different type of rods and the spirochetes. So, these are the first images which he developed after the microscopy of different specimens. The taxonomy of microbiology. Taxonomy is basically a branch of biological sciences, and in which we study about the three different interrelated sections, that is the classification, the nomenclature and identification. So, in this subject in taxonomy, the classification of microbes, the nomenclature of microbes and the identification of microbes is studied, and they are interrelated with each other. This taxonomy provides a system which is beneficial for all, such as single name or single label which is used for organisms, and that is common throughout the world. So, this system, this taxonomy minimizes the confusion which is made by names in the different areas, in the different regions, in the different labs, and allows to concentrate on important scientific issues and the phenomena, the processes which are related to these scientific issues. The classification, nomenclature and identification have a major and important role in the microbiology, and it provides the accurate and timely diagnosis of the infectious disease. So, this is an important part of the microbiology. The classification, the group of microorganisms that share same morphological, physiological and genetic characteristics. So, this is the classification. Microorganisms are classified on the basis of morphology, physiological and genetic characteristics, and this classification system is usually a hierarchic system, and this consists of the first one is kingdom, then it is classified into divisions, then this division is further classified into class, then that class is further classified into order, that order is classified into family system, that family system is classified into genus, and then genus is classified into different species. So, this is the classification system that is provided by the taxonomy. The species is the collection of bacterial strains, the different type of bacteria having same morphological, physiological and genetic characteristics that share similar characteristics morphologically, genetically, and as a group they differ from other types of bacterial species. So, these subgroups, there are some subgroups within a species, or they are subspecies, and these subspecies are divided into three different types: the biotype, the serotype and the genotype. So, when the species are classified on the basis of their physical characteristics, so these are called the biotype. If there are certain different antigens, certain different proteins, on the basis of proteins, if they are classified, then they are called the genotype, the serotype, and if they are classified on the basis of different genes, so that is the genotype of the species. The nomenclature, the naming of organisms according to the different rules which is established and the guidelines, this provides the acceptable labels by which the organisms can be recognized universally, and naming according to the binomial nomen system of nomenclature, in which microorganism is naming according to the genus and species. The genus is always capitalized and the species is never capitalized. For example, the Staphylococcus aureus, and all of these are written in italic or in underlined form. The Staphylococcus aureus, in this the Staphylococcus is the genus, where aureus is the species, or in short form it is written is the genus dot the species. The third part of the taxonomy is the identification, the process in which main and key features are defined, and then these properties as compared with the previously characterized organisms. So, there are two types of methods which are used for the identification of microbes. The first one is the phenotypic characteristics, and the second one is the genotypic characteristic. The phenotypic characteristics are those characteristics are the physical and observable characteristics of the organisms, and the genotypic characteristics are based on the genetic makeup, which including the nature of the genes, the type of the genes present and the nucleic acid which is present in them. So, on the basis of these two characteristics the organisms identified, and they are taxonomically classified. The phenotypic characteristics of different organisms, the phenotypic criteria, examples and their principles. So, on the basis of macroscopic morphology, these are the characteristics of the microbial growth patterns on artificial media, and when inspected with unaided eye. These include the size, the texture, the pigmentation of the bacterial colonies. So, the macroscopic characteristics include all of them. The microscopic morphology include the size of the organisms, the shape of the organism, the intracellular inclusions or the organelles which is present in the cell, what are the cellular appendages, the arrangements of cells which are observed with the help of a microscopic magnification. So, all these are the part of the microscopic morphology. The staining characteristics, the different organisms, microorganisms, they produce a specific particular color when they are reacted with the specific agents, and for this purpose a gram stain is very commonly used. So, bacteria are classified on the basis of gram staining into gram positive bacteria and gram negative bacteria. The different environmental requirements for the which are the part of the phenotypic characteristics, and the ability of the organisms to grow at various temperature in the presence of oxygen or other gases, at the various pH level, or in the presence of different types of ions, the different type of salts, the different types of any vitamins or any special nutrients. So all these have role in the phenotypic characteristics. The nutritional requirement of a microbe, in which the ability organisms to utilize various carbon nitrogen sources as a nutritional substrate when they grow under specific environmental conditions. The resistant profile of the organisms, if the microbe have specific resistance genes to specific antibiotics, or they have resistance to metals or toxins, so they are also a part of the resistant profile, which comes under the umbrella of the phenotypic identification. And the antigenic properties, which is established by the different microbes, having various serologic and immunologic methods are developed to find out that antigenic properties, and these are important and helpful to find out the relatedness among various microbial groups. And the subcellular properties, which is the establishment of molecular constituent of the cell that are typical of particular taxon or a particular group, and these are find out by various analytic methods, for example the cell wall components, the components of the cell membrane, or the enzymatic content of the microbial cell. So all these are the part of the phenotypic characteristics. The genotypic characteristics include the DNA base composition ratio and the DNA and RNA based sequence analysis. The DNA base composition ratio, it includes the 4 bases: guanine, cytosine, adenine and thymine. The extent to which the DNA from 2 organisms is made up is the cytosine and guanine. Related to their total base content can be used as an indicator of relatedness or lack. For example, an organisms with the high GC content more than 50% will not be closely related to any other organisms in which the GC content is less than 50%, less than 50%, or that is round about 25%. So both of these organisms are not closely related. And the second one of the genotypic criteria include the nucleic acid base sequence analysis. This is the order of the bases along a strand of DNA or RNA, which is known as the base sequence, and their homology or the similarity between the different organisms at their base sequence, that shows that they are either from the same group or they are either from the different group. So this was all about the microbiological history, the taxonomy, and the different divisions of microbiology, the scope of microbiology. If you have any question, you can ask. Thank you for watching. Assalamu alaikum. Allah Hafiz.